Let's present me a poster, Independent Component Analysis of Gene Expression Recapitulates Histopathological Properties of Pancreatic. In this work for analysis of transcriptomes, we use the convolution method called Independent Componental Analysis. In fact, we use its more robust implementation, Consensus ICA, proposed in this paper. The method separates a mixed transcriptome into statistically independent signals that could represent distinct cell types, biological processes, or technical biases. Mathematically, the method makes a matrix factorization, presents in origin gene expression E as a product of two small matrices independent signals S in the space of genes and weight M in the space of samples. Columns of matrix S show contribution of genes in each independent component's majority have no or minimal contribution. Therefore, we could use these contributing genes for functional annotation. Matrix M we could link to a clinical data. Some components could show predictive properties for a patient group of survival. For the survival analysis, we could combine scale weight of individual components with Cox regression log hazard ratio in order to get a risk score. For study of pancreatic cancers, we use three transcriptomic data sets, two public data sets of Peter Bailey and TCGA, and also dataset provided to us by German Cancer Research Center, by Jürgen Hacheisel's group. Next, we use two approaches. First, we analyze each dataset separately. The result could be compared by similarity of correlation between the identified independent signals, columns of S matrix in space of genes. Independent analysis of data allowed us to highlight the stable biological processes occurring in pancreatic cancer samples. Second, we combine these datasets. One is considered as training, another as testing, and run the analysis on the joint data. Here we somehow project the test data into a space defined by training data. After this, we could use machine learning models to be trained on ICA results and matrix from training samples and make a prediction about testing samples. Three datasets we first analyze independently, and components we link using R-square between columns of S matrix. Many components were found reproducible between datasets and formed a graph presented below. As shown in the graph, the individual components are highly correlated, which indicates the presence of common path physiological processes occurring in each dataset. For example, here we have cell cycle, many components from immune response and inflammation. Keratinization was a strong predictor of patient survival. Here you can see like components identified by ICA were annotated by biological functions and linked to survival on the patients by using Cox regression. Here you can see that secretion activity, gestation, and antigen binding reduce the risk of survival. There are no effects from immune response, gender, and exon development in the tumor cells, and different processes like keratinization, cell cycle, response to hypoxia, neoangiogenesis, activation of ERK signaling increase the risk of survival of the patient. As a proof that method works, we considered the tumor purity information that was assigned by histopathologists from the KFZ. On the figure A, you can see the weight of component linked to gestation, biological process with respect to a histologically identified and normal tissue fraction. You see that a clear increase on normal pancreatic activity. On the figure B, a strong correlation between one of the immune-related components and the percentage on inflamed tissue is presented. A very interesting profile is shown on the figure C that represents cell cycle component. You see that cell cycle grows with increases of tumor cell fraction until approximately 15%. Then there is a decrease that could be explained by a lack of resources for cell proliferation in a tumor core. The cell cycle component's behavior is also showed in a different considered tissues on the right. It was minimal for normal control, but already showed increase in the histopathologically normal tumor adjacent tissue and is high in tumors. That part of work was contributed this year to a project of proposal of my supervisor from Luxembourg, Peter Nazarov. In order to visualize how projection into independent components works, let's consider public datasets TCGA of pancreatic adenocarcinoma. The histological images into the dataset is available online as well. Here you can see distribution of TCGA samples in a space divided into independent components, cell cycle and digestion, which shows us a normal activity of tissue. Tumor tissues from samples with strong cell cycle components shows also a high degree of pleomorphism, high cell density and mitosis. In contrast, the sample with weak cell cycle and strong secretion-related component was almost normally histologically with real ductal structures and absent mitotic cells. Now, from the second approach, we combined two datasets. We used the KFZ data as training datasets and the data from Peter's Bailey paper as a testing. The log hazard ratio each from a Cox regression for the components was calculated from training set and combined with the component weight m and component stability r square. You can see the predictors of training data. We used 5-fold cross-validation on the independent testing cohort. Previously, we showed that pathophysiological processes affecting survival in the patient with pancreatic cancer are cell proliferation and keratinization. Consensus assay maps samples into the space of biologically mindful components. Also, ICA is able to characterize tumor purity. ICA of transcriptomic data from pancreatic cancer patients recapitulates the histopathological properties. In the future, we are developing a method that would link the convolved molecular profiles with histological features.